students, this is the third lecture of unit 3 biochemistry. In this lecture, we have to cover general reactions of amino acid metabolism and urea cycle and disorder of urea cycles and catabolism of different amino acids and their metabolic disorders. So, we will start with the general reactions of amino acid metabolism. Amino acids or proteins, they both are not stored in the body because there are totally turnover of proteins occurs in our body. Protein is metabolized to amino acid and these amino acids are again metabolized in body to give the different other compounds, and these proteins or amino acid undergoes different common reactions like transamination reaction, deamination reaction, decarboxylation reaction and the finally liberation of ammonia. Amino acid, you all knows that amino acid having amino group in their structure, carboxylic group in their structure. So, these amino group and carboxyl group is removed out from the this amino acid and the formation of ammonia or urea as an excretory end product. So, here in amino acid metabolism, urea is the end product, excretory end product of protein metabolism. So, this in this reaction, in this amino acid metabolism, the carbon skeletal, which is having in structure of amino acid, it is converted into the keto acid by transamination reaction. Why it is converted into the keto acid? To generate energy, utilized to generate energy for the formation of glucose, for the synthesized glucose, or converted into the formation of fat or fatty acids or ketone bodies. What are the general reactions of amino acid metabolism? There are three different metabolic reactions occurs in amino acid metabolism. The first one is transamination reaction. This reaction is reversible reaction. Here one amino acid is transfer its amino group to other compound and form keto acid, and the amino acid which is transferred to that particular compound is known as amino acid. So, here one compound is amino acid and the other compound is keto acid, and when there is transfer of one amino group from amino acid to keto acid to form keto acid to amino acid, and this amino acid is converted into the keto acid. This reaction is catalyzed by the transaminase enzyme ya amino transferase enzyme because there is transfer of amino group. Transamination reaction, transfer of amino group in this reaction is known as transamination reaction. This reaction is catalyzed by transaminase enzymes. There is no free ammonia is liberated in this reaction. In this reaction, only transfer of amino group from one compound to another compound, and this reaction requires peroxyl phosphate and a coenzyme which is derived from the vitamin B6. Okay? Next reaction is deamination reaction. In this deamination reaction, as the name says, deamination reaction, removal of amino group from the amino acid as ammonia, yani there is formation of free ammonia for the urea synthesis here. So, as the name says, there is removal of ammonia for the formation of urea. So, simultaneously this carbon skeletal amino acids converted into the keto acid, and this reaction occur here. This is oxidative and non-oxidative deamination reaction because deamination occurs in presence of oxygen and absence of oxygen also. So, in oxidative deamination reaction, amino acid is when reacted with the flavonoid mononucleotide and in water. So, in there is presence of amino acid oxidase here to form alpha keto acid, FNNH2 and ammonia. So, there is removal of ammonia from this reaction, and this FMN is again converted into the FMN H2 is again converted into the FMN because this is a work as enzyme here. So, one oxygen molecule is also used in this reaction for the formation of hydrogen peroxide. This hydrogen peroxide is again degraded with the help of catalase enzyme to form water molecule and oxygen is again released from this reaction, which is also used in this reaction. Non-oxidative deamination, where is no use of oxygen in this reaction, and this reaction occurs in those amino acid which has hydroxy group in their structure, like serine and threonine. And when serine is converted into the pyruvate here with the removal of ammonia and water molecule, and threonine is converted into the beta keto butyrate and removal of ammonia and water molecule in this reaction. So, it is commonly in this reaction there is removal of ammonia molecule, and serine threonine dehydratase enzyme is used in this reaction for the removal of ammonia. The third reaction is decarboxylation reaction. As the name says, decarboxylation or removal of carboxyl group in this reaction. So, this reaction give rise to physiologically active amines, and this reaction is catalyzed by the decarboxylase enzyme, which is having pyridoxal phosphate dependent enzyme. Pyridoxal phosphate forms a Schiff base here with an amino acid to stabilize a alpha carbanion, which is formed by the cleavage of bond between carboxyl and alpha carbon atom. So, there is beta keto acid is when reacted with the heat, so that this beta keto acid is having the this form enol compound, and one carbon dioxide is removed from here. By tautomerism, it is converted into the carboxyl group here, and removal of this ammonia in this this reaction. So, in this reaction, decarboxylation reaction occurs in those compound in which carboxyl group is present, carbonyl group is present in the in particular structure. If there is no presence of carbonyl C double bond O group in the structure, decarboxylation reaction does not occur, because in this reaction there is removal of carboxyl group from this amino acid. Okay. So, after removal of these after completion of these three reactions, transamination reaction, deamination reaction and decarboxylation reaction, we are shifted towards the urea cycle. In this urea cycle, this is a 5-step cyclic process hai, which is having 5 different enzymes, and in this reaction first 2 enzymes are present in the mitochondria, and the rest are localized in the cytosol. So, urea cycle is responsible for the formation of urea. Ammonia, which is freely formed from the deamination reaction, is very toxic in and it cause toxicity to our body. So, in our body this ammonia is transferred or converted into the urea, so it can easily move from the body via urine. So, in this urea cycle, the free ammonia which is synthesized or produced in the deamination reaction is utilized here in this urea cycle to form urea. The main compound which is formed in this urea cycle is urine here. So, this urea cycle having the 5 different steps with 5 different enzymes. In this, first 2 enzymes are present in the mitochondria, and the other rest 3 enzymes are localized or are present in the cytosol. The end product of protein metabolism is urea, and urea cycle is responsible for the formation of urea here. So, this nitrogen which is present, because we all know that amino acid having the amino group and the carboxyl group in their structure. So, nitrogen of amino acid which is converted into the ammonia is very toxic to our body. So, responsibility of urea cycle to convert all the produced or synthesized ammonia into the urea here. So, which is easily removed from the body by the urine, and this urea which is having the 80 to 90% of the nitrogen containing substance which is excreted in the urine, and this synthesized or located in the liver and transported to kidney for excretion of urine. This urea cycle is synthesized or located in the liver, but after synthesis of this urea it is transported into the kidney, and then they can excrete it via the urine. Okay. So, what are the steps? There are different 5 steps, as I already told you all that there are 5 different steps. In this, some steps are reversible, some are irreversible, but that depend on the particular the steps. So, first step is synthesis of carbomoyl phosphate. This is a irreversible step. Here carbomoyl phosphate synthase, which is an enzyme which catalyze the condensation of ammonia and carbon dioxide to form carbomoyl phosphate. The synthesized carbon dioxide in decarboxylation reaction and ammonia which is synthesized in deamination reaction are reacted here to form carbomoyl phosphate. This carbomoyl phosphate consumes 2 ATP and this is the main rate limiting step here, because if there is no formation of carbomoyl phosphate by free ammonia or free carbon dioxide, there is no formation of urea here. So, this is a rate limiting step here. Carbomoyl phosphate synthase first enzyme required N-acetyl glutamate for its activity. So, this it accept amino group from the free glutamine and it is does not require any N-acetyl glutamate for its activity. Okay. So, next step is formation of citrulline. This ornithine transcarboxylase enzyme is required for the formation of citrulline here. Ornithine, which is ornithine is although present in the urea cycle, it is regenerated in the urea cycle and again used in the urea cycle. It is present present in the urea cycle and this ornithine react with the citrulline, or these two are the basic amino acids and they are never found in the protein structure due to the lack of codons. So, these citrulline transported to cytosol by its transport system. And the third step is synthesis of arginosuccinate. Arginosuccinate synthase enzyme catalyze this reaction and it condenses to citrulline and aspartate to produce arginosuccinate here. This is second amino group of urea which is incorporated in this reaction. So, arginosuccinate, citrulline and before that carbomoyl phosphate, they all are important in this reaction. And the fourth is cleavage of arginosuccinate here. So, enzyme which is catalyze this reaction is arginosuccinase and arginine is the intermediate precursor for urea. So, arginine is required here as intermediate precursor. Okay. So, this fumarate liberated here. We all knows, if you have remembered, that fumarate is also a part of tricarboxylic acid cycle or Krebs cycle. So, fumarate liberated here is also used in that Krebs cycle. So, this is a connecting link between the tricarboxylic acid cycle and gluconeogenesis. In gluconeogenesis, if you all remember carbohydrate metabolism, gluconeogenesis is the synthesis of glucose from non-carbohydrate compounds. There is no carbohydrate compound utilized here for the formation of glucose. They are using ketone bodies, fatty acids for the formation of glucose. In such condition, when we are not taking diet, we are not taking food, at that situation gluconeogenesis is used or is required in the body for the formation of glucose. In the present when the body having fatty acids, ketone bodies in our body for the formation of glucose in non-diet condition, fasting situation, in such situation. Okay. So, the fifth step is formation of urea. The last step is urea formation. Here arginase enzyme is catalyzing this reaction, or ornithine enters here in the mitochondria. So, for its reuse for the urea cycle. So, here ornithine is already present in the urea cycle, it not converted from any compound. This is just present in the urea cycle for the formation of citrulline, and this ornithine enter mitochondria for again reaction or again formation of urea cycle or urea formation, and arginase is activated by the carbon dioxide and manganese. So, this is the main urea cycle. There are two different location or colors present here for the description of this particular urea cycle, that these two steps are not present in the these two steps are present in the mitochondria, and the other steps are present in the cytosol. So, these two enzymes are present in the mitochondria, and the rest 5 steps or rest 5 enzymes are present in the cytosol. So, this is the basically rate limiting step in the reaction where carbon dioxide and amino group or ammonia, which is synthesized in the amino acid metabolism, is transported in the mitochondria here, and in presence of carboxyl phosphate synthase first there is formation of carboxyl phosphate, and this step is the rate limiting step in this urea cycle. If there is not formation of carboxyl phosphate, there is not formation of urea, because this free carbon dioxide and this free ammonia is required here for the formation of urea. So, this is important question, and this is many times asked in the examination. So, which is the rate limiting step of urea cycle? So, this carboxyl phosphate synthesis is the rate limiting step in urea cycle. So, when this carbon dioxide and ammonia is reacted here to form carboxyl phosphate, and this carboxyl phosphate when reacted with the ornithine transcarboxyl enzyme. So, in this step there is also present a ornithine which is already present in the urea cycle, and this ornithine is reacted with the carboxyl phosphate here to form citrulline. This citrulline is and now this reaction occurs in the cytoplasm. There is now this reaction move from the mitochondria to cytoplasm here. So, this citrulline is synthesized in the cytoplasm. Here ornithine present in the cytoplasm and carboxyl phosphate present in the mitochondria, they are reacted together to form citrulline in the cytoplasm, and this citrulline is again reacted with the aspartate or arginosuccinate synthase enzyme, and from outer condition there is addition of aspartate enzyme to form arginosuccinate. Arginosuccinate is again reacted with the arginosuccinase enzyme, and there is formation of arginine. The one most important part is here, there is removal of fumarate here. When fumarate is removed here, this is a connecting link between the Krebs cycle or TCA cycle with this urea cycle. This fumarate is removed from this reaction and again reacted with the Krebs cycle to form other compounds, carbon dioxide, water there, and this arginine is synthesized in this urea cycle. This when reacted with the arginase enzyme in presence of water molecule. So, there is removal of urea here. So, urea is synthesized from arginine here in presence of arginase enzyme, and there is removal of or there is formation of urea in this reaction, and ornithine which is already present in the urea cycle is again reacted to the urea cycle, and there is formation of urea from this arginine, and this urea is from this liver to transported to kidney, so that this free urea can release from the body via urine. Okay. So, what are the disorders? So, this is total with the urea cycle, and now we are discuss the disorders associated with this urea cycle. There are so many rare life threatening genetic disorders which are associated with this urea cycle defect. So, why this disorder affect or why this disorder occurs? Because if there is no formation of urea in our body, there is no removal of toxic ammonia from our body. So, this can cause toxicity or life threatening genetic disorders. So, how we can control these things and how they can affect our body, because this is basically responsible by the defect in the enzymes. Because if there is no enzyme presence, particular enzyme which is used in the urea cycle, if they are not present in the body, they can cause disorders. So, these disorders of urea cycle basically shows life threatening effect, genetic disorders. It can start with the birth of the baby, and there is body is unable to break down. If there is no presence of enzymes in our body, so our body is unable to break down the waste product, so that there is no responsibility or no requirement or no there is impossibility for breaking down the protein to convert into the waste molecules, or removal of ammonia, removal of carbon dioxide from the body by the urea cycle. So, these disorders which are associated with the urea cycle is basically enzyme disorders. Enzyme disorders which is collectively known as inborn errors of urea synthesis or urea cycle enzyme defects or disorders. These the treatments of urea cycle disorders is generally restricted diet, protein restricted diet, so that protein is not converted to amino acid, or amino acid are जो है not metabolize in the body, but it is not possible, it is impossible कि we are not using the protein to rest of the life, but it is, that is why it is known as life threatening disorder. So, these are the various metabolic disorders. So, these are the various metabolic disorder. Hyperammonemia. In hyperammonemia, first the defective enzyme is carbamyl phosphate synthase first, that means that is the first step enzyme. So, product accumulated is ammonia, because ammonia is present in our body, but that cannot release. And hyperammonemia, first it is due to the ornithine transcarbamylase enzyme that is also responsible for the accumulation of ammonia in the compound. The second one is cetrolinemia, that is defection or defective of enzyme is arginosuccinate synthase, because responsible for the accumulation of citrulline in our body. And arginosuccinic aciduria disease occurs due to the defectivity or improper functioning or absence of enzyme arginase, so that the compound arginosuccinate is synthesized in the urea cycle, but it is not converted into the other compound. And arginonemia is a disease which is due to the defectivity or disorder of arginase enzyme, so that the arginine is the toxic compound which is present in our body, which is not converted into the urea. So, these disorders are very toxic, or they can show life threatening defect to our body. So, the next is catabolism of phenylalanine. Phenylalanine is an amino acid, and how to catabolize these phenylalanine. So, we have to discuss these. Phenylalanine, tyrosine and tryptophan are aromatic amino acid. They are having aromatic ring in their structure. So, biologically these are very important compounds, or many important compounds are produced from these amino acids, and deficiency of enzymes which are required for the metabolic pathway of these enzymes are responsible for the inborn error. These all are having inborn error, because enzymes are not present here for the catabolism of phenylalanine. And phenylalanine and tryptophan are nutritionally essential amino acid, because they are essential amino acid, they are produced from they are coming in our body by a diet. So, they are not having synthesized in our body. So, they are coming from the diet, and they all are glucogenic enzyme as well as ketogenic enzyme. And phenylalanine, in normal condition, degradation of phenylalanine normally occurs though tyrosine. So, phenylalanine, in presence of phenylalanine hydroxylase, and there is formation of tyrosine here in presence of dihydroxybiopterin reductase. Biopterin is synthesized here, and one molecule of NADPH is utilized here. And then the catabolism of tyrosine, considered together, the metabolism of phenylalanine and tyrosine considered together, this tyrosine is again this tyrosine is with the help of tyrosine aminotransferase enzyme and converted into the para hydroxyphenyl pyruvate, and this is converted into the homogenatizase. If this is not if tyrosine aminotransferase enzyme is not present, this may cause tyrosinemia second, and para hydroxyphenyl pyruvate deoxygenase enzyme is not present, this may cause tyrosinemia second. Catabolism of tyrosine also, this homogenizate is again converted into the methyl acetoacetate, and this is converted into the fumaryl acetoacetate. This if these enzymes are not present, or they are working defectively, they cause alkaptonuria, tyrosinemia, and they can cause the inborn error to our body system. And disorders of phenylalanine metabolism, phenylketonuria, this is due to the deficiency of hepatic enzyme phenylalanine hydroxylase, and this may cause defect in the mental retardation. Normally patient cannot walk properly, and growth of patient is also affected. Phenylketonuria is detected by the Guthrie test, and it is basically depend on the plasma level of the phenylalanine. Albinism is due to the lack of synthesis of pigment melanin, because this is also synthesized by the enzyme phenylalanine and tyrosine. So, this is also albinism is also due to the absence or deficiency of catabolism of phenylalanine and tyrosine enzyme. And alkaptonuria is an enzyme disorder because of homogenatase oxidase. It accumulates in the tissues and blood and excreted in the urine. This may cause an inborn error or life threatening effect. Tyrosinosis or tyrosinemia type 1, this is due to the deficiency of the enzyme fumarate acetoacetate hydroxylase and mannanyl acetoacetate isomerase. The next is tyrosinemia type 2, which is also known as Richter-Honard syndrome. This syndrome is occurs due to the enzyme tyrosinase transaminase enzyme. This is not, if this enzyme is not present in the body, this may cause tyrosinemia type 2. So, this is all over for today's lecture. Thank you.